Hello and welcome to another video lecture from Mr. Watkins at mrwatkins.com. This particular video is on protein synthesis using the model, and this is going to be on the transcription part. If you haven't watched the other videos, you should do that before you watch this particular one. This one is an interactive one, and so you'll need to or it'd be best to download the pieces that you need for the model, and those pieces are one that's the protein synthesis, simulating the process, and with that particular download there should also have been a couple of sheets. That's the student answer sheet. So these are the things that the procedure on how we're going to do this, as well as the answer sheet that gets turned in later. You also will have a DNA molecule sheet, this big sheet here, and you'll also have a messenger or a RNA nucleotide sheet, and that's what this one is here. So first thing you need to do is get this particular model, and we've got to get ready to do this simulation. So you need to cut this piece out, and you just cut around the outside edge, and then notice this little dip right here, that needs to be cut out. Once this side is cut out, it gets placed at the bottom of this other side, so that you have a nice big long chain. And once you have that big long chain put together, and here's one side of it, once you have it put together, we're going to go through the next process about breaking those hydrogen bonds and separating out the piece of DNA to begin the process of transcription. Okay, so we need to break those bonds, but before we break the bonds here, we need to put in the complement. We need to basically do the base pairing, but put in the complement here. And so I've got thymine here. This is DNA, so this is also going to be adenine, cytosine to guanine, adenine to thymine, and you don't have to turn them sideways. You can just do it as you wish. So there's thymine, we got adenine, thymine, guanine. So just fill it in. It's on the tape, it's not going to work. Get it all filled in. And again, the assumption is made is that you have read and seen the other two videos that deal with protein synthesis. So this is just to reinforce it. So now that we have our piece here, we're going to go ahead and break this hydrogen bond. Now with DNA replication, the thing that broke the hydrogen bond was DNA polymerase. In protein synthesis, what breaks the hydrogen bond between the two nucleotides is RNA polymerase. So I'm going to get some scissors, and I'm going to simulate the breaking of the hydrogen bond between the two strands using these scissors, and it is done by polymerase. Now RNA polymerase is an enzyme, and that enzyme basically speeds up that chemical reaction. RNA polymerase really does about 3-4 different things. Remember, in DNA replication, we had a special molecule, helicase, that split it. We had a special molecule, DNA polymerase, that added on the nucleotides. In protein synthesis, RNA polymerase is what does a lot of that. It breaks the bond. It base pairs by adding in the nucleotides. It joins those nucleotides together. It closes up and zips back the molecule, the DNA molecule. So it's kind of almost like this special machine that goes right down the piece of DNA and makes the cuts, puts in the new piece, and basically does the entire process of copying. And it is a little more complex than that. So I'm going to simulate what's going to happen first, which is the breaking of the hydrogen bond here, and I'm just going to cut straight through. And please note, it says you need 2 strands, so that means you need to cut all the way through till there is nothing more to cut, all the way through to get your 2 strands here. So here's one of my strands, and here is the other strand. Those are the 2 strands. I've just cut through and broke that. Now, transcription, excuse me. So we have basically unzipped this piece of DNA. So if you notice, the DNA before was together. We broke the hydrogen bonds, and we have unzipped the DNA and getting it ready to be read by the RNA polymerase. And so the next piece then is excuse me, the next piece then is to base pair the RNA nucleotides onto this piece of DNA. So that means you're going to have to come out and get your RNA, and what I would suggest is you just go ahead and cut them all out, and I'm going to do that right now. I'll cut out all of these pieces, and then we'll come back in and we will base pair the DNA polymerase, or excuse me, the nucleic acids to the DNA. All right, so now we've cut out all our RNA nucleotides, so here's one of them, and we need to start the process of base pairing. Remember, RNA polymerase is actually what's doing this, but we're going to simulate its method of action. It's already unzipped the DNA. So here we go. Thymine always bonds with adenine. That's its complement. So I'm just going to put a little piece of tape right here to bind it together. And then adenine bonds to thymine in DNA, but it actually is going to be bonding to uracil. And that's because in RNA, there isn't any thymine, it is uracil. Now, one of the things you're going to notice here, as I move my hands back out of the way, is that we are going to get a little overlap here, and that's just part of the model that we had put together here to help you understand the process of transcription. And so this process will continue, and you'll continue to add nucleotides all the way through until you get to the very end here. So each time we're going to add a nucleotide until we get to the very end on the other side, the other end of this molecule. The other thing that happens is that the bond between the phosphates and the nitrogenous base also occurs, and again, because it's overlapping so much, you really can't see what's going on. But once we get done with the very end of this, we're going to run a piece of tape down that, and that's what it means by taping the nucleotides together to make one long strand, because that's what we're really wanting to do. Is we want a big long strand here of the mRNA. That's the message from this piece of DNA. So I'm going to go ahead and finish out the rest of putting these things together so that we can then cut that off. Alrighty, I've taped all the mRNA nucleotides, the nucleotides on here, just as it says, and then I've also put a piece of tape here to represent that these things are now not just bonded this way to the piece of DNA, but they're also bonded to each other, and so we have this big long strand here. Please also note that when I did the bonding, I made sure that these things lined up pretty close, right opposite each other, and we did have some overlap. This is important because there's this reading frame that's in the ribosome. The ribosome, in translation, in the next video, is the one that lines up the codons, the three bases that are on the mRNA that have a code for a particular amino acid. It lines them up in those groups of three. And so if you don't put it right across from each other, in other words, you don't maintain the structure, because the structure is just as important as the type of molecule there, then the process doesn't work properly. So our next step now that we've done this, we've got our copy, and we know what sequence this is in, we know we've got the right distance between the bases. The next thing that happens is that this piece of mRNA, this is DNA, here's mRNA, is removed, and so you cut the mRNA from the piece of DNA. Now some of the students go, well, why didn't I just tape this all together and just sequence one right after another? Well, that goes back to this idea that the piece of mRNA has to be not just the right sequence, so that's why we tape it to this to get the right sequence, but it also has to be the right distance. Otherwise, it just doesn't fit into the ribosome, and you've got to have it fit in the ribosome. That's what's going to read it. So here's my piece of DNA. Here's my piece of mRNA that I made off of that piece of DNA. And so I'm going to take this mRNA and I'm just going to set it aside for now. And remember, before we started this whole process, this complementary strand had been pulled apart so that we could get to this sequence and we could make our copy. Now, this piece of DNA needs to zip back together, needs to go back together. We've made our copy here. We've got used this, to make our copy, and now we've put this piece back, and so we need to tape this molecule back together here so that we now have our DNA. It's the sequence on the inside is nice and safe and has been restored to the double strand that it once was prior to doing the model, prior to cutting it with the RNA polymerase, cutting it in our case with the scissors to simulate that. So I'm just going to zip this thing back together. I'm going to tape it back together so that these two pieces stick together and we're done. Now, we don't need this piece of DNA anymore. This piece of DNA, we've copied our recipe. We now have our message. So we really don't need this piece of DNA anymore. Please look on the handout. There's a thing that you're supposed to do with this DNA if this is going to be for a grade for me. So here's my mRNA that we had just made. In organisms, particularly eukaryotic organisms, the nucleotides, these things here, are a code for a particular protein. RNA polymerase, when it reads it off of this piece of DNA, here's the piece that we read it off of, when it reads it, it just reads and puts the nucleotides in where it's supposed to go. But in some genes, there is an area that may not be read. And this area that's not read is called an interon. It's an intervening sequence. And by not read, I mean out in translation, it's not needed to make the protein. So this mRNA is what we call an immature mRNA. It needs to have these interons, these intervening bases that don't belong there to make the protein we want. Those need to be removed. Now, some people believe that these little chunks are junk and they're not, they don't have any other function. There is some growing studies that show that some of this is not correct. Take AP and IB bio and we'll get into more detail about what that actually means. So the stuff that's going to be removed, we need to do that. The pieces, though, that of the bases in the sequence that actually goes onto the ribosome and then is converted, is translated, is expressed into the amino acid sequence, those are called exons, and those are going to be the ones that remain. So on our model here, we're looking at the sequence here. We're going to read from 5 to 3, so A, U, G, A, U, A, and so on. We're looking for a sequence, and we just made this up, but this is the sequence, that's the interon. We're looking for the sequence that is going to be CCGGC, and that's what this is right here. So there's another enzyme, and again, AP and IB bio, you'll get into all the details of that, but this sequence needs to be, this stuff needs to be pulled out. So there's an enzyme that actually physically comes in and cuts off that piece of, or that bad piece of DNA, this intervening sequence, and so we're going to physically come in and cut out this intervening sequence here and physically just take it completely out of our mRNA, because it is not expressed. So we've just removed it. And then we need the sequence together because this is what needs to be read. So we're going to stick the two pieces back together and glue them together, or tape them together. And the enzyme responsible for this actually does this process. It finds these sequences that are not part of the end product. They're not going to be part of the protein that we need to make, so they're intervening, they're in the way, and so we need to remove those and just leave the sequences that will be expressed, the exons, and so we have done that now. Now that we have this all done, this is our sequence. This is the exon that's going to be used, read by the ribosome, to make the protein that we need to make. And then this piece here, this interon, if you're following along with the model, needs to be put onto the sheet, and then you'll just follow along with the student answer sheet, and that's where you need to put this. This is the end of RNA, making the mRNA, the end of translation. This piece, this mature piece of mRNA with its interon removed, only exons remain, is then sent to the cytoplasm so that it can then be used to make, or at least get translated into the amino acid sequence. You need to watch the next video on translation, and that will pick up from where we left off right here.